I only recently transformed from a grad student to a postdoc, but it might not come as much of a heat shock to you that I've already had a number of transformative experiences. So let's talk about bacterial transformation using chemically competent cells and heat shock.
So transformation is when we stick a piece of genetic information into bacteria. Often this genetic information is in the form of a circular piece of DNA called a plasmid. And we can put in the plasmid instructions for like making a protein and get the bacteria to make the protein for us. We can also get the bacteria to just make lots of copies of the plasmid. So this is a really key technique in molecular biology and biochemistry and all of these different methods. And one of the main ways that we do it that we'll talk about is using this heat shock with these chemically competent cells. So basically you take these cells that are weakened and then you use heat to open up pores in the membrane and allow the DNA to get in. And then you help it heal, and then you can do whatever you need to do with it. And so we'll talk about how you can plate it to use the antibiotic selection. So these plasmids have an antibiotic resistance gene that you can then use to select for the cells that actually have the plasmid. And so we'll go into all of this in more detail. But a little technical note, so transformation is a form of transfection. So transfection is when we stick genetic information, so in the form of DNA or RNA, into cells. And when we do this into bacterial cells, we call it transformation. But we can also stick in, genetic information into other types of cells, and when we do this, we call it transfection. And so we avoid that word transformation because it has other uses and connotations, such as things with like cells turning cancerous. And so we don't want to confuse that sort of thing. And so we call it transfection typically in other cellular contexts. But when we're talking about bacteria, we call it transformation. So I'm going to talk mainly about this chemically competent cells and heat shock, but there are other delivery methods for transfection. Another common one is electroporation, where you kind of use electricity to open up pores in the membrane and create a charge gradient to help the DNA get in. There are also ways that you can use like cationic carriers, such as these polymers such as PEI, which are positively charged, and they're going to help neutralize the DNA, which we're going to be talking about something similar when we're talking about the calcium chloride, but here we have a long positively charged thing. And there are also lipids. So like the mRNA vaccines say, those have this lipid nanoparticle. And so these often are these like cationic lipids so that they have a charge too. And so we're not going to talk about these as much, but this is just giving you an overview. And then there are also viral delivery methods. When a virus is introducing genetic information, we typically call that transfection, but we're going to be talking about this transformation. So as I mentioned, what we're typically transforming is a plasmid, and this plasmid is going to have often some sort of insert, which has the genetic information for making a protein or making some sort of other genetic information that we want the bacteria to either just make lots of copies of or to actually make a protein out of. And the plasmid is also going to have an antibiotic resistance gene that's going to allow this cells that take up the plasmid to grow in the presence of that antibiotic, whereas cells that don't have the plasmid will not grow. And so this is going to be an important concept, is this idea that not all the cells are going to take up the plasmid. And if you clone that plasmid, I'm using a cloning method like restriction cloning or SLIC, there's a bunch of different methods, but often the cells have to, like not all of the clones are going to be successful in terms of actually getting the plasmid piece back together. And the bacteria is also going to have to do some work in some of these cases to actually heal the DNA after the pieces are put together. And the bacteria kind of has to do the stitching. And so for these sort of things, not all the cells that take up that like kind of partial plasmid, that's like plasmid that's not quite finished yet, not all of them are going to be able to totally finish the job. And so you're going to have a lower transformation efficiency. So less of the cells are going to actually transform and take this up, and then less of them are actually going to have a functional plasmid, and this is going to make it so that you're going to get less colonies. And so each of these colonies that you're going to get on your plate is going to be a genetically identical group of bacteria. So basically an original bacteria that had that plasmid, that functional plasmid. It's going to take root and grow on top of itself, and you get these globby colonies, and then you could do various things with them. So just make sure that they actually have the plasmid, that the plasmid sequence is right, and then actually make proteins and stuff. And so you can stick these plasmids into different types of cells, as we talked about in other posts. You have like cloning cells where you're just going to make lots of copies of the plasmid, as well as expression cells. So the cloning cells would be things like these Th5 alpha cells. And then for expression cells, you might have things like BL21. Fun note, DH5 alpha, so these common cloning cells that we use, the DH comes for Douglas Hanahan, and he actually developed the protocols for these calcium chloride heat shock that we're going to talk about. So let's talk more about it. Okay. So the basic overview of how this works is that we're going to take these deep frozen chemically competent cells, and we'll talk more about how we make them chemically competent. But these are some weakened cells. And then we're going to mix them with DNA, our plasmid DNA. And then we're going to use heat shock to open pores in the bacterial membrane. Then we're going to do an outgrowth step where we're going to add bacterial media, so the bacteria food, in this case without antibiotics, to let the bacteria recover. Once they've recovered, we're going to plate them on agar dishes and then get colonies. So in terms of a practical procedure, you're going to have the heat shock. Typically, you're going to do it in some sort of like water bath or a thermal cycler, or not thermal cycler, but like a thermal heat block. And this is going to be done for like 40 seconds at like 45°C. We'll get more into the details, but it's going to vary depending on your tube type and cell type. Then you add the media, you stick it on this little shaker, and then later you're going to plate it out. And we'll get much more into all of these details, but let's talk about what's actually going on at the biochemical level. So this is basically the situation that we're facing. We have this plasmid we want to get into the bacteria, but the bacteria has things that are going to make this difficult. So if we look at a bacterial cell, it's such as E. coli. So what you have here is you have the cell, and you have the interior of the cell, the cytoplasm, the bacteria's own genome, and then you have this these cellular membranes and cellular walls. And so basically, there's this inner membrane which has these phospholipids. These phospholipids are similar to the phospholipids that make up our cells. And these phospholipids are amphiphilic, which means they have a part that's hydrophilic, so a part that water likes, in this case, these hydrophilic negatively charged heads, and a part that's hydrophobic, so that water avoids, these hydrophobic tails. And the hydrophobic parts are going to kind of blob together. The hydrophilic parts are going to be exposed to the water, and you get this lipid bilayer. And so then the outer membrane in bacteria, so the bacteria have these inner membrane and an outer membrane, and their outer membrane is also going to have these lipopolysaccharides. So this is different from our cells. They have these lipopolysaccharides. Importantly, these are also negatively charged. So you have this very negatively charged membrane. They also have this in bacteria. You have this like peptidoglycan wall and this like periplasmic space in between them. Crucially, there are points called zones of adhesion where the membranes are fused. So remember, we have these two membranes, and there are these parts where they're fused, these zones of adhesion. So they have little tunnels already, but it's hard for the DNA to get into through these tunnels because the DNA is too big. So these tunnels are pretty small, the DNA is too big, and the DNA is negatively charged. And so if we look at the backbone of DNA, we have this negatively charged backbone, these phosphate groups. If we look at the membrane of the DNA of the bacteria, we have a lot of negative charges as well. And you know that like charges repel. And so if we want that DNA to get close, we're going to have to give it some help. We're going to have to help neutralize those charges. And we can do this by adding a positive charge. So we can add calcium chloride So calcium chloride, this is a salt form. So a salt is when you have a neutral combo of a positively charged thing, so a cation, and a negatively charged thing, an anion. So in calcium chloride, you have CaCl2 because calcium has a +2 charge. Chlorine has a -1 charge, so you need two of them to neutralize the calcium. But when you dissolve them, these are going to come apart. It's going to dissociate, and so you're going to get those calcium ions, and those calcium ions can then hang out with the DNA, and then they can hang out with the membrane, and they can help them get close together. And so this is, as I was talking about, this is similar to the idea was when we use cationic carriers like these long PEI molecules to help wrap up the DNA and coat the charge. But in here we have this calcium chloride, and we use this calcium chloride to like coat these cells. So when we prepare these competent cells, typically, so you can buy the cells pre-prepared, or you can make your own. And you make it by like soaking them in calcium chloride and helping them. This is also going to like destabilize their membranes, as well as, so that's going to like weaken the membranes as well as make it easier for the DNA to get into contact. Okay, so what's going to happen is that once the DNA is in contact, so you're going to mix the DNA with the cells. And so often this is done with like 1 to 10 nanograms of DNA with 50 to 100 microliters of competent cells. We'll get more into handling the competent cells later because you have to treat them with much TLC, but like including individual aliquots. So you'll have these individual aliquots with like 50 to 100 microliters of competent cells. I typically use like 50 microliters. And so then what you want to do is you mix the DNA, and you're going to do this on ice. So then you take that mixture and you're going to stick it in the heat bath. And so you don't want, when you're doing this, you're not, because the cells are really fragile, you don't want to vortex it, you don't want to mix up and down. Instead, just give it a gentle finger tap, or swirl it with your pipette tip. And then you are going to go to the heat shock step. And this is where you're going to take that tube and you're going to dunk it into the hot water bath. When I say hot, it's really only like 42 degrees Celsius. So the exact details are going to depend on your various protocol. Typically, I do about a 40-second heat shock. The actual times and temperatures are going to vary depending on your tube size and type and the cell type. And so look at the protocol for the ones that you're using. But what's going to happen when you do the heat shock is there's a variety of mechanisms at place, and there's not completely understood exactly what's going on. But a few of the things that are going to help the DNA get in. One is that when you heat shock, it's going to help widen the pores in the cell membrane. So those zones of adhesion. It's going to help it kind of shed some of the lipids, make these pores bigger. So now there's a route for the DNA to get in. But the DNA has to kind of like want to get in. And so how do we make it want to get in? The heat's going to help us with this too. When you open the pores, it's going to depolarize the cells. The bacteria, it keeps its inside of the cell. Typically, it keeps it more negatively charged by pumping out protons, so pumping out like H+. And it's actively doing this. And so then when you open these pores, you're kind of disrupting that whole system. So the bacteria, now those protons can flow back in. You get the cell, the inside of the cell is going to become less negatively charged. And this is going to allow the negatively charged DNA to find the cell more attractive. And this DNA is also going to have more energy to move because you have the heat. So when you have heat and molecules have more energy, they can move around more. So you might think, okay, well, can't the DNA just move right back out of the cell as much? And so, but actually, because the DNA, when the DNA is out here, it's not going to be insulated. When it's in here, it's going to be more insulated from the heat. So you're going to get this thermal imbalance where the free DNA out here is going to have more heat, more energy, and go into the cell. So as I mentioned, the electroporation is another method that we can use to get the plasmid into the cells, but in this case, we use electricity. And how these cells are prepared is going to be a little different. So instead of calcium chloride, you actually want to remove all of those salts. It's going to prevent like arcing and stuff. And then you can use electricity, this electroporation method, to get things into the cell. Notably, electroporation can also be used for other cell types. And so this chemically competent transformation with E. coli is really, really helpful. But if you want to use a different type of cell, you might need to use a different method, such as electroporation or one of those other transfection methods if you're using the more complicated types of cells. Okay, so once you get to that heat shock, and now you're going to move on to this outgrowth cell, this outgrowth step. So what do I mean by this outgrowth? This outgrowth is basically where you're going to take those cells that have taken the DNA in, and you're going to let them heal with a little growth in the absence of the antibiotic. And so at this point, we're going to have those cells that we treated with electroporation, they're going to join in here as well. And so we're going to add the media, so the cell food without the antibiotic, and we're going to let it shake in this little shaker. Typically, you can add, in terms of media, you can add either like LB or SOC or SOB. So LB is lysogeny broth. It's kind of like this classic broth that we use as classic bacterial media. And it has this basic nutrients that the bacterial needs to survive. We can also, especially if you're dealing with something that has a lower transformation efficiency or that sort of thing, you can use SOC, the super optimal broth with metabolite repression. What you can see here is that it has more glucose. It has glucose, sorry, whereas traditional LB doesn't. And so this also has more nutrients. This is going to allow it to have more nutrients it needs to help heal and grow. And so you can get the cells to grow more before you plate them on your antibiotic plates. This can help if you have a low transformation efficiency to have more of the starting material. But typically I just use LB. Okay, so the outgrowth step, the amount that you're actually going to outgrow it is going to depend on what antibiotic you're using. So antibiotics, basically the antibiotics are going to be really important because we're going to use them to allow only the cells that take up the antibiotic to grow. But the cells have to actually be able to make the things that make them resistant. So the plasmid has the instructions for making the antibiotic resistance product, and the cells have to actually be able to make that. The problem comes with some of these antibiotics is that how some of these antibiotics work is by actually preventing the bacteria from making proteins. So common examples of these protein inhibitors are like tetracycline and other tetracyclines, and kanamycin and other aminoglycosides. So both of these are going to target protein synthesis. If you're preventing them from making proteins and they need to make the antibiotic resistance protein, well, now you're preventing them from making the thing that they need to prevent the cells from making the things that they need. So instead, you want to give them time in this outgrowth step to actually make the proteins, in the absence of the antibiotic. So this outgrowth is going to be less crucial if you're using antibiotics that target other mechanisms in the bacteria, such as ampicillin and other beta-lactams that are going to target the cell wall synthesis. So typically for like ampicillin, I usually do like a 30-minute outgrowth, whereas for tetracycline and kanamycin, I often do like an hour long. You can also, sometimes you might even need a longer outgrowth, such as when I'm doing my baculovirus subcloning, when I'm making this Plasmid. It has these like multiple antibiotics and things that it needs, as well as a more complicated, it has to heal the plasmid that I put in after doing this like cloning procedure. And so it needs that time to heal, and it needs that extra time to heal. And so typically I do like at least 4 hours or overnight to actually allow it to do this. Okay, so after that outgrowth step, it's going to be time to plate. And the amount that you're going to plate is going to depend on kind of the transformation efficiency that you're expecting. So I typically plate like around 50 microliters for an intact plasmid. And for one of those cloning reactions like I was talking about, where the cells actually have to heal the product, or even for a ligation product where it's not going to be 100% effective beforehand, you might want to plate more. And you also might want to transform more of these when you're doing when you're actually mixing the DNA with the cells. In the case of these slick products, what I typically do is I actually want to plate all of it. So Now, when I added the media after the heat shock, I typically add like 800 microliters, somewhere between like 800 microliters and a mil is typical. And so you can't plate that whole volume on one of those little plates, or it would just be like a soup, just like a soup. So instead, what you want to do is, I would say, like a max of like 200 microliters probably that you want to plate. So what I do if I want to do all of it is I'll just like spin it down in the microcentrifuge, give it a couple minutes spin, remove all the media, add fresh media, so like 100 microliters, resuspend the cells, so like pipette up and down to resuspend it, and then plate all of that. And this will allow me the maximum chance of getting those colonies. But if you were to plate that much of a pure plasmid, like a mini prep or something that you put in there, then you're going to have like just a lawn of bacteria. So you're not going to have individual colonies. You're just going to have like colonies everywhere, and you won't be able to like pick individual ones. So when you have a mini prep and stuff, the DNA is already healed. So it's already just like this full circle, and it's often going to be super coiled. And this is going to make it really good for getting into these cells. And so they're going to have a high transformation efficiency. Speaking of which, if you're having problems with your transformation, you're not getting many products, or if it's a new batch of competent cells, what you might want to do is do a positive control. And so you can do a positive control. This is showing a control for cloning, but you can do a positive control for your transformation on the competency of the cells by taking a plasmid that is already full. So sometimes, often these competent cells, like if you buy them or something, they'll actually come with a control plasmid. So basically any plasmid will work. Just make sure that you're plating it on the antibiotic that has antibiotic resistance for the antibiotic that you're using. And then basically this will tell you that you should definitely get colonies for these. So if you don't, then there's something wrong with either your procedure or with your competent cells themselves. So let's talk about how we can treat those competent cells to make sure that they actually remain competent. So as I mentioned, these cells are very delicate and you need to treat them with TLC. So when we do those chemical things to make it chemically competent, you're weakening the membrane. So this is great for letting the DNA in, but it also is going to not so great for actually working with these cells, because now they're going to be really fragile. So if you can buy, when you buy them, you can sometimes have like these like one-shot things where they're single aliquots. Other times they'll come in single tubes. If they just come in a single tube, you're going to want to aliquot them out the first time you thaw them. If you're prepping your own, you're going to want to aliquot them when you're making them. It's important to make these single-use aliquots because every time you freeze thaw them, you're going to kill a lot of the cells, and you're going to reduce the transformation efficiency, and make them like non-competent. So then you're going to store these aliquots in the -80 freezer. When you're working with them, you want to keep the tubes on ice. So this includes when you're doing the labeling and stuff, and when you're adding the DNA. After you do that heat shock, when you take it out of the water bath, you want to stick it into the ice. And then add the media, typically when it's on the ice, before you then move it to your shaker. And so when you're handling the tubes, you want to make sure that you're handling the tubes from the top and not from the bottom. So don't put your like warm hands on the bottom of the tube when you're dealing with it. You're like trying to open the lid and that sort of thing. Instead, you want to really just work from the top. You might want to pre-loosen the lids and that sort of thing before you actually go to add the DNA, so that you're not like at crunch time having your pipette like moving all over the place when you're trying to keep things fairly sterile. But make sure that you're handling from the top of the tubes and not holding them at the bottom and thawing them out and heating them up and making them unhappy. And we already talked about how you don't want to vortex or you don't want to pipette mix. Instead, just like mix by gently shaking, swirling it with the pipette tip or tapping it with your finger. And then, as I mentioned, you want to make sure that if you're having trouble, you might want to do some sort of positive control to make sure the cells are still okay. And again, the exact details are going to depend on the type of cells that you're using, the type of DNA that you're transforming, the type of tubes that you're using. So there are a lot of different things. And so it's important to look at the actual protocols for the cells that you're using, for the tubes that you're using, and that sort of thing. But this is just a general overview of the technique, as well as some typical typical like parameters and stuff for when you're doing this. So hope it helped.